going to look at how we can regulate protein activity, still using a conformational change, but using two different common mechanisms. One is covalent modifications, and we're going to look in particular at something called phosphorylation. And the second is binding and hydrolyzing of nucleotides, focusing mainly on GTP and GTP binding proteins, but we can also see that ATP is hydrolyzed to cause movement of proteins, things like motor proteins. I'll show you that through a video. So to think about how these nucleotides can affect protein structure, it helps to think about what the nucleotides themselves look like. So here is ATP, which you probably have heard of as an important molecule for energy usage within the cell, right? This is what mitochondria are producing. You take the stored energy in glucose and rip it apart through the processes of glycolysis, citric acid or Krebs cycle, and then electron transport and oxidative phosphorylation. And that oxidative phosphorylation is what produces this molecule, ATP, right? And which we have the adenine, the ribose, and then the three phosphate groups. And this is what we're focusing in on for phosphorylation. This last phosphate group can be removed from ATP and transferred onto particular amino acids of proteins and covalently bonded to them. And we're going to see that this covalent bond of an inorganic phosphate group onto a protein is going to change its shape. ATP, we're going to say, donates a phosphate group, and this causes a conformational change. So then the question is why? So you can maybe get some answers if you focus in on the inorganic phosphate and look at the characteristics. Remember, the characteristics of amino acid side chains were important to folding of the protein. The characteristics we saw were charged, polar, or nonpolar. What we can see here is we have two negative charges. So that's going to be important to how adding a phosphate group causes a rearrangement of shape in a protein. So in this image, they're trying to show you what I think is somewhat of a subtle shape change in the particular protein. The protein is in green, and the green ribbon represents the primary structure, the sequence of amino acids. And they focus in on a serine side chain. And it's this particular amino acid side chain to which a phosphate group is going to be covalently bonded. So they're magnifying that particular serine side chain. And we can see we have the non-phosphorylated version and then the phosphorylated version of that serine side chain. The source of the phosphate group is ATP, adenosine triphosphate. When we remove one phosphate, we get adenine diphosphate and one inorganic phosphate that has been transferred to the protein. And this causes a shift or change in shape. This change in shape is here in this end of the protein, right? If we want to return the protein back to its original shape, you remove the phosphate group and it goes back to its original shape. Phosphorylation is controlled by two different families of enzymes. First is protein kinases, which transfer the phosphate group from ATP to the protein and create that covalent bond. The second is protein phosphatases, which cut the covalent bond holding that phosphate group, which is shown magnified here and simplified here. They remove that phosphate group. So protein kinases add the phosphate, phosphatases remove them. As you add or remove the phosphate, you're switching or toggling between two different protein shapes. And again, the reason why you're going to get a change in shape is because of those negative charges. This is going to cause a rearrangement of the interactions of the amino acids. If there is a positively charged amino acid side chain nearby, that would draw to the serine, bring them closer. If there is a negatively charged amino acid nearby, right, you would have repulsive forces pushing them further apart. Things like that are going to cause a shifting or a rearrangement in protein shape. In this image, we're looking at regulation of a protein, like an enzyme, through a kinase and a phosphatase. The circle image is trying to show you the inactive conformation of the enzyme, and this would be the active conformation. In this case, adding the phosphate group activates the enzyme and it's turned on. Removing the phosphate group inactivates it again. So again, in this case, the kinase is activating the enzyme. There is no rule that phosphorylation activates, so keep that in mind. Phosphorylation causes a change in shape. Sometimes that may activate an enzyme, but other times it may be that the kinase and phosphorylation of that protein inactivates the enzyme, whereas removal of the phosphate group from the phosphatase activates it. So in this case, which enzyme, the kinase or the phosphatase, is activating? It's the phosphatase. In reality, most proteins that are phosphorylated have multiple sites where they could be phosphorylated. One phosphorylation site could be activating, another phosphorylation site could be inactivating. So it's really dependent on the particular site. What is constant is that kinases add phosphate groups, phosphatases remove them, and this addition and removal of phosphate groups causes conformational change. So here's a question: how can proteins be regulated? Just looked at enzymatic activity of proteins can be regulated through covalent modifications. So phosphorylation is a covalent modification. And then there are some other covalent modifications as well. They can not only change or toggle an enzyme between an inactive or an inactive site, they can work on proteins that aren't enzymes and affect things like binding partners, what proteins are stuck to. Proteins are often working together in large complexes, so it may be that the complex won't assemble or come together unless you have phosphorylation or other covalent modifications in the correct site. Proteins can also vary according to their location within the cell. For example, if you have a transcription factor, something that's regulating transcription, and it's in the cytoplasm, it's not active. It doesn't matter if the shape is optimal for regulating transcription. If it's in the cytoplasm, it can't do that. It needs to be in the nucleus. Also, some proteins may switch between a location within the cytoplasm and the cell membrane. Another important way of regulating the protein is its presence. Presence can be regulated in the positive or kind of production section where through transcription and translation, and those are important aspects of protein regulation, but they also can be degraded or broken apart. And there's another type of tag called ubiquitin, which is a covalent modification that tags a protein to be destroyed in the proteasome. For me, I think of a proteasome like a wood chipper. So the protein is like a full-length stick, right, that goes in, and then you put it through the wood chipper and all the little pieces come out. The protein is like that, goes into the proteasome, and then amino acids or small peptide chains are what come out. So all of these activities can be regulated by covalent modifications. I decided to keep this older picture from an earlier version of your textbook to show how these might work. Picture is showing you a long stretch here of amino acids. So the green line or bar is amino acids. And then they're showing you some different types of modifications. Phosphorylation, there's 1, 2, 3, 4 phosphorylation sites. So 4 different amino acids can be phosphorylated. There's a site where you can have acetylation. And acetylation is common in proteins like histones and is important in regulating transcription. And here's that ubiquitin, which can send proteins to the proteasome. Each individual protein could have multiple covalent modifications. The covalent modifications are added by signals received from the cell. So the cell receives signals and it can cause phosphorylation at any of these 4 sites, and or acetylation, and or ubiquitination. The idea being that you could have a single phosphorylation site, you could have 2 phosphorylation sites, or you could have phosphorylation and acetylation. You can have different combinations of these covalent modifications, which can then lead to different outcomes. Depending on which particular covalent modifications you have, the protein might bind to proteins in a complex, proteins Y and Z, or move to the nucleus, or be degraded or ripped apart in the proteasome, or go to the plasma membrane. And these are in addition to if this were an enzyme being activated or switching between an inactive and an active form. So these covalent modifications and the particular combination of modifications on a particular protein affect its activity. So this is a form of regulation. The last thing we're going to cover in Chapter 4 is another form of protein regulation, which is binding to a small nucleotide. This one is GTP, guanosine triphosphate. And this is somewhat similar to phosphorylation, but there really is a difference. And sometimes students get these confused. So I'm going to try to make the differences clear. We are still looking at a nucleotide that has negative charge. However, this is not a covalent modification. We will start at the bottom here with an inactive GTP binding protein, this green circle here. What you can't see, and maybe I'll draw in, is that there is a binding site within this protein. And that binding site fits a guanosine nucleotide, GTP, guanosine triphosphate. To switch from this inactive conformation to what is here the active conformation of the GTP binding protein, GTP is going to come into that binding site, and it's going to bind through those weak non-covalent bonds, hydrogen bonds, electrostatic interactions, things like that. So it is not covalently bonded to the G protein. And this is just a regular binding event. There is no kinase working here. Okay, so sometimes students want to put a kinase here. There is no kinase working here. Protein starts off as inactive. If the site is empty, the binding site is empty, there is a lot of GTP in the cell, high concentration. If it's open and available, GTP will come in and bind and activate the GTP binding protein. We need to also be able to inactivate the GTP binding protein, and this happens through the process of GTP hydrolysis. We are losing a phosphate group, but the phosphate group is being removed from GTP and leaving behind the inorganic phosphate. The extra one is just released, and then the protein is still bound to guanosine di or 2-phosphates, and it's inactive in this GDP bound form. When it's bound to GDP, when there's no guanine nucleotide in that binding site, it's inactive, or when there's a GDP. So this protein needs to release the GDP, calling it dissociation, so leave the binding site, which creates an opening, and then GTP can come in and bind that site and activate the protein. And the reason we get a change in shape is, again, GTP is bringing in lots of negative charge on those phosphate groups, and that's going to cause a rearrangement in the shape of the protein. So I've added these notations here to tell you what I just said. For starting at the inactive form, GTP binding causes a conformational change that activates the G protein. So this is our active form. G protein is inactivated when GTP is hydrolyzed to GDP. So this is an enzymatic reaction that is happening within the G protein itself. It has that ability to cut off the phosphate group, and then the protein must release the GDP from the guanine nucleotide binding site. It becomes kind of free form before GTP can bind again. I have a little notation here that this happens very fast. There's plenty of GTP around, it's easy to bind, but the dissociation is slow. So it turns out that there are some enzymes that are going to help with this process. Shutting down this active G protein or inactivating it would be very slow on its own. But there is a class of proteins called GTPase activating proteins, or GAPs, that associate with the G protein, and together, when it's in that gap-bound state, it accelerates or speeds up GTP hydrolysis. Similarly, after GTP hydrolysis, when you're bound to a GDP, diphosphate, that's a slow process. It can be accelerated by binding to proteins that function as GDP-GTP exchange factors, or GEFs. So these GEFs are going to help this G protein to release GDP from the binding site and kind of start off as that blank slate where GTP can bind and reactivate. So we get a cycle of activation and inactivation with GTP binding proteins. Note that there are 3 different states here: bound to GTP, bound to GDP, and bound to neither. This is different than what happens with the kinase and phosphatase, where there are 2 different states: covalently attached phosphate group or not. We'll put aside this type of regulation for a little while. Phosphorylation and the activity of G proteins are both going to be important in Chapter 16 when we look at cell signaling.